Good morning, students. Let's begin today's class with an interesting topic. We are going to learn about the basics and overall process of genetic engineering, a major area of research in the field of modern biotechnology. As we all know, biotechnology involves the manipulation of organisms and their components to make useful products. To begin with, what is the difference between these three mice depicted in the picture? Well, the middle mouse is a normal mice, whereas the other two are genetically modified to express a green fluorescent protein. Such genetically modified mice are used in disease detection using bioluminescent imaging to identify different types of cells. Similarly, another important example, many years ago, diabetics needed pig or cow insulin to be injected into their bodies for their sugar control. This was not an ideal option as it caused issues like allergies, high cost, small yields, immunological rejections, and so on. Today, the insulin the diabetics inject is human insulin and is produced by microorganisms such as E. coli and yeast. But how do these microorganisms produce this human insulin by genetic engineering? To answer this, scientists are able to turn certain microorganisms into factories that improve our health and environment. Coming to what is genetic engineering? First, let's get into the basics of the DNA. An organism's DNA, as illustrated in the image here, makes up its genes which codes for all the protein an organism needs to survive. Each gene codes for a different protein or a part of the protein. Genetic engineering involves the manipulation or changing of the DNA of an organism. Genetic engineering or recombinant DNA technology, as defined here in this slide, can be termed as the artificial manipulation of DNA or altering the genes of a living organism to produce genetically modified organisms. In short, they are called as GMOs. The figure here depicts an organism which has the foreign DNA, which is shown here in the yellow color, integrated into its genetic material. Now this is termed as a GMO. Such GMOs have practical applications in medicine, agriculture and industry. The next slide tells us the different terminologies used for genetic engineering. Genetic engineering is referred to by other names such as gene cloning, rDNA technology, gene manipulation, gene splicing, and gene modification. Now let's move on into the two main objectives of genetic engineering. There are two purposes of genetic engineering. First, sometimes we require large quantities of pharmaceutical proteins to be made, as depicted in the figure here. So scientists use transgenic bacteria to produce large volumes of these pharmaceutical proteins such as insulin, growth hormones, or vaccines. The second purpose here, as illustrated in the second image here, an organism B can have a gene introduced from an organism A to make the organism B have some advantage that A has naturally. Now the organism B is termed as a GMO or transgenic organism. Let me clearly explain this to you with a suitable example. A gene that produces a toxic chemical in organism A, such as bacteria, can be transferred to organism B. In this case, in the figure, the maize plant in the picture. The maize plant is now made insecticide resistant by the process of genetic engineering. It is now protected against the insects that eat it, especially the grasshoppers and the caterpillars. Let's now look into the three kinds of genetic modifications that are possible. First, inserting a foreign gene from one species into another, as a result forming a transgenic organism. Next, altering an existing gene or its products is changed, or finally by altering gene expression levels. Thus, by the process of rDNA technology or genetic engineering, changing the genome enables engineers to give desirable properties to different organisms. At this juncture, it is important for you all to know what is meant by a recombinant DNA. As defined in the slide here, recombinant DNA refers to the creation of new combination of DNA segments that are not found together in nature. Recombinant DNA are also termed as chimeric DNA. They were developed by two scientists, namely Boyer and Cohen, in the year 1973. Can you see the image here? This depicts the fusion between two genes to form a recombinant DNA. One gene here is the human gene. The second gene here, shown in the purple color, is the bacterial plasmid DNA. They are fused together to form the recombinant DNA. Now let's move on to the history and milestones of GMOs. To begin with, in the year 1969, two scientists, Stuart Lynn and Werber Arber, discovered restriction enzymes in the bacteria E. coli. Next, in the year 1972, the first recombinant DNA molecule by Paul Berg, utilizing restriction enzymes and DNA ligases, were created. Next, in the year 1973, the first genetically modified bacteria were formed. Next, in the year 1974, the first genetically modified mice were created. A big breakthrough in the year 1977 was the DNA sequencing technique invented by Frederick Sanger. The first commercial development of GMOs began in the year 1982. Sales of genetically modified food began in the year 1994. Another important breakthrough in the history of GMOs was in the year 1996 by the birth of the first cloned animal, Dolly the sheep. In 2003, uh, begin to sell GMOs as pets. Example, glowfish. Glowfish was sold as pets in the market. Finally, in the year 2015, first genetically modified animal to be approved for food was a salmon. Till date, there are numerous breakthroughs and numerous research that are happening in the field of GMOs that still need to be explored further. Next, let's move on to the tools used in genetic engineering. This picture here depicts the most important requirements to execute a genetic engineering experiment. Primarily, enzymes such as restriction enzymes and DNA ligases are required to perform the experiment. Now, what are these restriction enzymes? A restriction enzyme or a restriction endonuclease or a restrictase is an enzyme that cleaves DNA into fragments at or near specific recognition sites within molecules known as restriction sites. Restriction enzymes belong to a broader class of enzymes known as the endonucleases. They act as molecular scissors in the process of genetic engineering. The next important enzyme is the DNA ligases. DNA ligase is a DNA joining enzyme, also called as a molecular glue. The DNA ligase catalyzes the formation of covalent phosphodiester linkages which permanently join the DNA pieces together. Subsequently, the next important requirement is the foreign DNA or gene of interest that should be isolated from the target cell for the experiment. The third requirement are the vectors. These are one of the most important requirements in gene cloning experiments. Vectors are generally DNA molecules. The commonly used ones are the plasmids and viruses that are used as a vehicle to carry a particular DNA segment into a host cell as part of a cloning or recombinant DNA technique. A vector typically assists in replication or expression of the inserted foreign DNA inside a host cell. Next, we move on to the next requirement, which is a host cell or host organism. Host organisms into which the recombinant DNA is introduced. The host is the ultimate tool of recombinant DNA technology, which takes in the vector engineered with the desired foreign DNA with the help of enzymes. Widely used host cells in genetic engineering experiments include E. coli bacteria and eukaryotic yeast cells. Finally, as you can see in the image here, culture media, buffers and reagents are also a part of the experiment. A commonly used example of genetic engineering to understand the overall process of genetic engineering in detail is the production of the human insulin using E. coli bacterial cells as factories. Now, why use bacteria as factories? Now, let me give you two important answers to this question. The first advantage, they have extra chromosomal DNA in addition to the circular DNA. This is called the plasmid DNA. Look at the figure here. Can you see? This is the bacterial E. coli cell. Here, the bacterial chromosomal linear DNA is seen. Next to that, it has the extra chromosomal DNA, which is termed as a plasmid. Another advantage is that bacteria are unicellular and reproduce asexually and produce quick clones. This picture here illustrates E. coli bacteria and a human cell with a nucleus, and within the nucleus we have the DNA and a gene that produces the insulin gene. The first step in the recombinant DNA technology involves the isolation of the plasmid DNA and the isolation of the foreign DNA or insulin DNA from the target cell. Then in the next step, they are going to be cut with the same restriction enzyme to generate complementary sticky ends. These enzymes act as scissors, molecular scissors, to cut the DNA at specific recognition sites termed as the restriction site. In the subsequent step, the insulin gene, can you see here? The insulin gene, that is the gene of interest, and the plasmid DNA is joined together, or you can term it as ligated together by the enzyme DNA ligase. The ligase catalyzes the reaction by sealing the sulfur phosphate backbone of the DNA. Now we have the recombinant DNA, which now has to be transferred into the host cell by the process of transformation. See the figure here, students. This is the host cell. This is the recombinant DNA. And now this recombinant DNA is going to be transferred into the E. coli host cell by the process of transformation. Bacteria is then allowed to reproduce and produce multiple clones. Clone of identical bacterial cells now produce large quantities of insulin. Can you see here? These are the clone of identical bacterial cells. Now they produce large quantities of insulin. The insulin thus produced by this method is now isolated from the clone of bacteria using special techniques. I hope all of you have understood the basics of genetic engineering and the detailed process of genetic engineering experiment. In the next class, we will discuss about the applications of genetic engineering in the various fields. Thank you all for your attention.